DNA cloning. I'm P. Sai Pranati. Introduction. DNA cloning is the process of making multiple identical copies of a particular piece of DNA. The insertion is done using enzymes that cut and paste DNA, and it produces a molecule of recombinant DNA or a DNA assembled out of the fragments from multiple resources. What is the point of making many copies of a DNA sequence in a plasmid? In some cases, we need lots of DNA copies to conduct experiments or build new plasmids. In other cases, the piece of DNA encodes a useful protein and the bacteria are used as factories to make the protein. Steps of DNA cloning. DNA cloning is used for many purposes. As an example, let's see how DNA cloning can be used to synthesize a protein. The basic steps are: cut open the plasmid and paste the genome. This process relies on restriction enzymes. In DNA ligase, insert the plasmid into bacteria, use antibiotic selection to identify the bacteria that took up the plasmid. Cutting and pasting the DNA. How can pieces of DNA from different resources be joined together? A common method uses two types of enzymes, restriction enzymes and DNA ligase. A restriction enzyme is a DNA cutting enzyme that recognizes a specific target sequence and cuts the DNA into two pieces. Our goal is cloning is to insert a target gene into a plasmid. The plasmid, which has a single cut site, the target gene fragment, which has a cut site near each. Bacterial transformation and selection. Plasmids and other DNA can be introduced into bacteria such as harmful E. coli used in labs in a process called transformation. During transformation, specially prepared bacterial cells are given a shock. This encourages them to take up foreign DNA. Not all colonies will necessarily contain the right plasmid. That's because during a ligation, DNA fragments don't always get pasted in exactly the way we intended. Protein production. Once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria. The bacteria serve as miniature factories churning out of large amounts of protein. Once the protein has been produced, the bacterial cells can be split open to release it. Protein production. Once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria. The bacteria serve as miniature factories churning out of large amounts of protein. For instance, if plasmid contained the human insulin gene. Use of DNA cloning. DNA molecules built through cloning techniques are used for many purposes in molecular biology. Biopharmaceuticals. DNA cloning can be used to make human proteins with biomedical applications such as insulin mentioned above. Gene therapy. In some